Okay, so today we're going to focus on organic compounds or biomolecules. Remember, whether it's written down or said, they are equally important. So you cannot just write down, your notes cannot just be what you see written on the screen. Remember, if it is spoken and said, especially more than one time, you need to be sure to be writing that down as well. Okay, so let's talk about the difference between an inorganic versus an organic compound. What makes something an inorganic compound or an organic compound? Okay, so inorganic compounds, they do not have a carbon-hydrogen bond. So they may have carbon in them, but they are not, that carbon is not going to be bonded to a hydrogen. For example, these would be things like water. Water doesn't have any carbon in it, so that's pretty obvious that's going to be an inorganic compound. Carbon dioxide is an inorganic compound. There is no carbon-hydrogen bond there. There is carbon bonded to two oxygens, but not bonded to a hydrogen. So that makes it an inorganic compound. Your organic compounds then do have that carbon-hydrogen bond. And we have 4 basic categories of these organic compounds. Okay, so we're going to be looking at carbohydrates or sugars, and we're going to look at each one of these individually. Okay, lipids, also known as your fats. Okay, proteins, and nucleic acids. And so our focus today is going to be each one of these different categories. We're going to look at structure and function. And unfortunately with this particular topic, there's not a lot to understand here. The information is relatively simple, but there's a lot to memorize here. So you're going to need to start putting the time in to memorize what these different molecules do and what they look like. So our first category of organic compounds that we're going to look at are the carbohydrates. Okay, and so some particular characteristics of carbohydrates is that they only contain carbon, hydrogen, and oxygen. So they will not contain any other elements. They will not contain any nitrogen, any sulfur, any phosphate, only carbon, hydrogen, oxygen. So when you're looking at pictures of these different organic compounds, if they only contain carbon, hydrogen, oxygen, that can help narrow it down for you. Also, they will contain it, that carbon, hydrogen, oxygen will be in a particular ratio, okay, of a 1:1 ratio, with that being carbon, hydrogen, and oxygen. For example, over here on the side, this is a molecule of glucose over here. And if y'all remember your chemical formula for glucose, it is C6H12O6. And if I reduce these numbers down, that's a 1:1 ratio. So if you saw just a chemical formula, for example, let's say you saw a chemical formula that looked like this. C24H48O24. That should help you narrow that down, that is a carbohydrate. It contains only carbon, hydrogen, oxygen, and it contains it in that 1:1 ratio. Okay. Also, we're talking about our carbohydrates. A lot of them will end in the suffix "-ose". Okay. "-ose" means sugar, so our carbohydrates are sugars. They are things like glucose, maltose, galactose, fructose. We're going to talk about these more, each of them. Sucrose. But again, you may, in a question on a test or a quiz or a lab, come across a name of a compound that you don't know. But if it ends in this "-ose" suffix, then that means you need to be thinking that it is probably a carbohydrate. Most of these organic compounds are going to have what is called a monomer and then a polymer version of the organic compound. So the monomer, this is our building block. These are our subunits. Think if you had a giant box of Legos. You know how they have the Lego sets that make a particular, a castle or a boat or something like that. Let's say you have a set of Legos that makes a castle when you assemble them. The monomer would be each individual little Lego. It's the building block. It's the individual single unit. Okay, that would be our monomer. Whereas your polymer, okay, so the other version of this would be your polymer, and that's going to be made of lots of monomers. So that's going to be your finished boat or castle. It's all these monomers hooked together. Okay, if you think about what these words mean, mono means one, poly means many. So the monomer is the individual little subunit. The polymer is lots of those monomers put together. So in a carbohydrate, so monomers and polymers are not specific to carbohydrates. You will have monomers and polymers in carbohydrates, you will see them in proteins, and you will see them in nucleic acids. So the monomer of carbohydrates specifically is what's called a monosaccharide. So this is our monomer for carbohydrates or sugars. Okay, this saccharide here should cue you in that you're talking about sugars. So a monosaccharide is going to be our building block of our larger polysaccharides in carbohydrates. You have three basic monomers for carbohydrates: glucose, fructose, that's the sugar that you find in fruit, as well as galactose. And these monomers are going to have a ring shape to them. If you remember your picture of glucose just a minute ago, and I'll bring it back for you, okay, it is shaped like a ring. Okay, and that should cue you in that you are talking about a carbohydrate. When you see this ring shape, it may not be 6, it may be 5, okay, but that lets you know that that is a carbohydrate. So the monomers of carbohydrates are monosaccharides. You have 3 of them: glucose, fructose, and galactose, and they will be shaped roughly in a ring shape. You are going to be asked to identify these molecules by what they look like. So you have to be comfortable with the pictures of these molecules. So if your monomer of carbohydrates was a monosaccharide, then your polymer of carbohydrates would be a polysaccharide. Remember the saccharide sugar, poly is many, many sugars. So these polysaccharides, they are going to look like a bunch of rings hooked together. Okay, because there are many of these individual monosaccharides linked together. And so again, that should be another clue as to what you're looking at when you see these long chains of rings hooked together. If you look at this picture here, this is an example of cellulose. That's one of our examples of a polysaccharide. Okay, cellulose, remember it ends in that ose, letting you know again it's a carbohydrate or sugar. Cellulose is what you find in plants. Okay, it is it's a structural component for plants. It's what's in the cell wall of the plant cell. So that tells you that one of the functions of carbohydrates is to help with structure or support. Okay, another example of a polysaccharide would be glycogen. Glycogen is the storage form of glucose in animals. So for instance, that's how we store our extra sugar or glucose that we take in our food. We link it together in the form of glycogen to store it. Okay, another one would be starch. Starch is another storage. This is how plants store their extra glucose. So all 3 of these are examples of polysaccharides. So for monosaccharides, you had glucose, fructose, and galactose. For polysaccharides, you have glycogen, starch, and cellulose. Okay, and again, you see your individual rings of each of the monomer here linked together to form the big polymer. So now that you've seen a polymer and a monomer example, let's take a second to talk about the reactions that form a polymer or either break a polymer apart into its monomers. So if we look at this picture up here on the top left, so we're over here now, okay, this is what's called a dehydration reaction. Okay, and so a dehydration reaction is the synthesizing or the building of the polymer. So I went from the short polymer, so the forming polymer here. So let's say I'm talking about carbohydrates here. I've got a bunch of glucose hooked together and I'm trying to add more of that on there. And I'm going to add this monomer to that short chain polymer. And I'm going to do that with a dehydration reaction. And so it's called a dehydration reaction because I've lost water here. And that's what happens when you dehydrate, right? You're losing water. So when I remove that water, that allows those two molecules to hook together. So all I need you to know right from now is basically the name of these two reactions. That a dehydration reaction actually links molecules together. So dehydration reactions make polymers. So that's how you would make glycogen, starch, and cellulose. Okay, it's by dehydration reactions. And then a hydrolysis reaction, hydro is water, and lysis is to break or to burst. Okay, and so a hydrolysis reaction uses water to break a polymer apart. So a dehydration synthesis reaction is going to make the polymer, where a hydrolysis reaction is going to break the polymer into its monomers. Because remember, the polymer is made up of those little pieces. So that's, for instance, when we are exercising and we need more glucose to fuel our muscle cells, we use hydrolysis reactions to break the glycogen apart so we can have those little pieces of glucose that it's made up of. So now that we know what these monomers and polymers look like, let's talk about what they do for carbohydrates in particular. So our primary function of carbohydrates is it's an energy source. And in particular, it's the short, immediate energy needs. Okay, so carbohydrates are a source of energy. And from now on, you'll just see energy written as a big, large E. Okay, so that's your carbohydrates' number one function, is to provide that quick, fast source of energy. Glucose would be the molecule that would help you do that. It does it for plants, does it for all organisms. Second function of carbohydrates would be energy storage. So this is what those polysaccharides can do. We're talking about glycogen and starch. Glycogen was energy storage in plants, in animals, where starch was the energy storage in plants. And a third function of our carbohydrates is that they can offer structure and support. Remember that polysaccharide cellulose that we saw a picture of and we talked about? That offers support in a plant cell as it's in the cell wall of the plant, and so almost acts as the skeleton for the plant. So moving on to our next category of organic compounds, we're going to go to the lipids now. Lipids are our one category of organic compounds that are not going to have a specific monomer and a specific polymer. For example, in carbohydrates, we had monosaccharides. All those monosaccharides are the different kinds of monomers for carbohydrates. We don't have that for lipids. Lipids have a huge range structurally of what they look like. So let's look at some examples of them so you can see how different they all look. So for example, what I have up here in the top left, this is a fat molecule. In particular, it is what's called a triglyceride, because it's made up of, on the left here in this beige, okay, that's a glycerol molecule. It's made of one glycerol, so that's where that part comes from. And then it's made of 1, 2, 3 fatty acids. That's where the tri part comes from. Okay, and so each one of those is what's called a fatty acid. Okay, so down here at the bottom in the left, this is the one we're going to talk about tons and tons, especially when we get to cells, is what's called a phospholipid. And a phospholipid, okay, it has a phosphate group up here at the top. It has a phosphate head. And it has, again, these fatty acid, what are called these fatty acid tails. Okay, so that's what our phospholipid would look like. Up here on the right, okay, is what's called cholesterol, which is actually a steroid hormone. So we can have lipids that are hormones, okay, steroids. They work a little bit differently than some of our other kinds of hormones. Okay, you'll notice this one has the rings in it, similar to our carbohydrates with their structure. Okay, some other examples of lipids we don't have pictures of here would be oils, okay, waxes. So we've got bunches and bunches of different examples of lipids. We've got fats, okay, in particular triglyceride. We've got phospholipids, steroid hormones, showing you the picture there of cholesterol, oils, waxes. All of them are different kinds of lipids. If you look closely at the pictures, though, one of the things you may notice that they all have in common is lipids do contain carbon, they contain hydrogen, and they contain oxygen. Some of them may contain phosphate, for example, with the phospholipid, but all of them will contain carbon, hydrogen, oxygen. They do not have that nice ratio that carbohydrates do. They usually have a way more carbon and hydrogen associated with them than that oxygen. For instance, if you look at your fatty acid chains, which you see in a couple of these different examples, these are just big, long chains of carbon and hydrogens together. And that's important for the function of the lipid, which is energy storage. That's where they store it, is in those big, long carbon-hydrogen chains. So some characteristics of lipids is that they are what we call non-polar, which means they have no charge to the molecule. And by charge, I mean positive and negative charge. There is no electrical positive or negative charge to the molecule. And because they have no charge like that, because they are non-polar, then they are water insoluble, which means that they do not dissolve in water. Think about salad dressing. If you have salad dressing in your refrigerator, any kind of oil, vinegar type salad dressing, it will separate and you have to shake it up before you can use it. And that's because the oil, which is the fat or the lipid part, does not mix with the vinegar because the oil is non-polar, but the vinegar is polar. And so you have to shake them up to get them to mix up because they don't, the oil, the lipid, will not dissolve in the vinegar. Because lipids were so structurally different, they have a lot of different functions. Okay? Structure and function go hand in hand. So one of the functions of a lipid is that they can be a chemical messenger. These would be the hormones. Okay? So steroids, cholesterol, they're not all bad. Okay? We have both males and females have steroid hormones in them. They work a little bit different than the protein hormones that we have. Okay? But they still carry messages from cell to cell. Okay? Another function of lipids would be they make up the cell membrane. Those phospholipids that we saw a picture of, okay, that's what the bulk of our cell membrane consists of. And in between those phospholipids are some cholesterol molecules. So they make up the cell membrane, so they control what goes in and out of their cell membrane. And they also act as energy storage. Mentioned that on the last slide. Those long chains of carbon and hydrogen help store energy. Okay, and so the lipid molecules that do that would be those fats and triglycerides. And let's take a quick look at how those are classified and move on to proteins. Okay, so fats can be classified as either saturated or unsaturated. Okay, remember a fat is an example of a lipid. Lipids contains fats, oils, waxes, steroid hormones, phospholipids, a lot more than just fats. Okay, but fats in particular can be classified as saturated or unsaturated. Saturated fats have no double bonds, which means there is no extra carbon-carbon bond. It is always carbon, carbon, and then bunches of hydrogen around them. Okay, so there's no double bond there between the carbons. These are going to be solid at room temperature. Okay, so these are things like butter, bacon grease. And you know, if you've let a bacon grease pan sit there and cool, okay, it will become solid. So these are also usually animal-based. Our unsaturated fats, okay, unsaturated fats then, they are going to have at least one double bond. And what that does is that double bond makes these fatty acid tails, it makes them bend. You can see it right there, it bends. And so because it bends, they can't be solid at room temperature. They can't get close, the molecules can't get close enough together to be solid at room temperature. So these are liquid at room temperature, and these are usually plant-based. Olive oil, that would be a good example of an unsaturated fat. Okay, we have two categories of organic compounds left. I know this is a long video, especially for your first one. Okay, hang in there. You need to get used to it, and you need to make it through all the way. Be sure you're taking notes of things that are said as well as written. And remember, it's a video. Pause it, take a break, come back, finish it up. Okay, but be sure you get all the way through. Okay, so our next one we're going to talk about is proteins. Okay, proteins, we're going to go back to that monomer-polymer structure. So proteins are going to have a monomer. They're going to have a building block. The monomer of proteins is going to be amino acids. This is crucial. This is just a fact you have to memorize. Proteins are made up of amino acids. They're not the same thing. The amino acids are the little pieces that make up the big, long protein. Okay, your amino acid has a particular structure to it that's going to be important to you for enzymes, for DNA. So we're going to take a look at that structure really quickly. So amino acids, this would be an example of amino acid here, they have this pattern. They have this NCC pattern. So if you're looking at, remember I told you you're going to have to be able to recognize these pictures. If you're looking at pictures and you see this NCC pattern, then you need to think amino acid. If you see it repeating NCC, 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 then you need to be thinking protein because you've got a lot of amino acids together. Okay, every amino acid, so this is my amino acid down here, every amino acid has an amino group and a carboxyl group, and these are the same for every amino acid. Okay, they never change. They're the same. They contain this amino nitrogen group and that carboxyl group with the double bond to the O and the alcohol group there. What's different is what's called this R group or the side chain. Sometimes it's called the variable group. Okay, that changes. That is what makes each amino acid different. And so that R is like an X in algebra. It can stand for anything. It could stand for a long chain of carbons. It could stand for some sulfur. It could stand for some phosphate. That R is variable. It can change from amino acid to amino acid. Our polymer of proteins then is a protein. So in carbohydrates it was a little bit different. Our polymer was the polysaccharide. In the protein, the polymer is a protein. Okay, so it's going to be lots and lots of amino acids. And that protein is going to consist of amino acids held together by peptide bonds. In the carbohydrate, we didn't name the bond that holds them together, but here we're going to. These are called peptide bonds in between each of the amino acids. And that's important because a protein is sometimes referred to as a polypeptide. You need to be comfortable with both of those names. So the polymer is a protein. Protein can also be called a polypeptide because it has all these peptide bonds within it. Okay, so let's take a look at the picture of the existing protein, and maybe that will help some with that. So you can see your polypeptide forming here. Okay, so you see each one of these amino acids, I'm going to make this nice and big and cover up some of the words. Okay, so you see each one of these amino acids here. Okay, so each one of these guys is an amino acid. So I'm linking this third amino acid on there. Remember, I join my monomers together by using that dehydration synthesis. So there goes my water out of my reaction. So I did my dehydration synthesis and I formed a peptide bond. This one here is also a peptide bond, holding each individual amino acid together, okay, to then make the polymer or the protein. Protein structure is very specific as it's getting to the polymer stage. Okay, so what we're looking at here is a overview of protein structure. And again, this is a protein. So we're looking at the polymer here, or the polypeptide. So I've got lots of little amino acids hooked together. So the protein has initially what's called its primary structure. And so the primary structure is just going to be the order of the amino acids. Each amino acid has a name. You can see that here: valine, glycine, serine, leucine. You're not going to be expected to memorize their names, but it is helpful that you realize they have a name. So that's all that the primary structure is what order those are in. If I switch up that order and I make it serine, glycine, leucine, okay, now I've got a different primary structure. The secondary structure right here has two forms. What's called either the alpha helix, so that little symbol there stands for alpha helix, or beta pleated sheet. So that's what that kind of capital B there stands for. That's our secondary structure. Okay, and so this is just the twisting or folding of the amino acid chain. Protein function is based almost completely on the protein's shape or structure. If the protein changes its shape, if it changes how it was folded, you can see all these crinkles and folds in this next step here. If it changes or breaks some of those crinkles and folds, that protein no longer works. So the order in the folding of this is extremely important. So the secondary structure is when those amino acids start bending and twisting. Then we get to what's called our tertiary structure. So this is our third step of the structure. So we had our primary, our secondary, and our tertiary structure. The tertiary structure is when it folds into a 3D structure. So instead of, and I'll show you an example of this happening in class, which I think will help a lot. Okay? Imagine that you have a piece of paper. Okay, and you take that piece of paper and you fold it like you were folding a fan. You know, when you were a little kid and you made the little accordion fan. Okay, that would be your secondary structure. That would be your beta pleated sheet. Now you took that accordion fan and you crumpled it up into a ball. Now you've made it into your 3D tertiary structure. Okay, so there's lots of folding happening here. And then some proteins have a fourth step, which is what's called our quaternary structure, and that's only how they interact with another protein. For example, in your body, you've got hemoglobin. Hemoglobin is 4 proteins. So each one of those individual 4 proteins went through a primary, secondary, and tertiary folding, and then the quaternary structure is how the 4 of those stuck together. So every protein is not going to have this because every protein is not going to stick together with another protein. Like I said, I'll model this in class, and I think that will help a lot for you to see it. Right now, though, I just need you to understand the basics. This is what primary is, this is what secondary is, tertiary, and then quaternary. So some functions of proteins. So proteins have a wide variety of functions. They also can act as hormones. Remember, hormones were our chemical messengers. So they can help take information and pass it from one part of your body to another part of the body. Plants have those hormones. Okay, proteins are also important for transport. They do this a lot in the cell membrane. You'll see proteins embedded in the cell membrane that will help move things back and forth across the cell membrane. I mentioned hemoglobin on the last slide. Hemoglobin helps transport oxygen throughout your body, so they act as a transport structure. Proteins also have immune functions. Okay, our antibodies that help fight off foreign invaders, they are made up of proteins. Okay, proteins also can have structural, protective type functions. Things like your fingernails, your hair, okay, cartilage, all of those are made up of proteins. And so those all offer support and protection to the rest of your systems. Okay, one more thing that we need to talk about with proteins is a specific kind of protein, which is called an enzyme. We've got your next video, which is going to be a lot shorter than this one, is going to be on enzymes specifically. Okay, but they are a type of protein, and what they do is they speed up chemical reactions. Without enzymes, we would not be able to work. Okay, that RX in there is reactions. So enzymes are a special type of protein that speed up chemical reactions. Like I said, we're going to do a whole video on enzymes next, and it's going to be half as long as this one. Okay, you've made it. We're on our last category of organic compounds, this one being nucleic acids. So our monomer of nucleic acids is what's called a nucleotide. Okay, a nucleotide has 3 parts to it. You're going to need to know all 3 parts. Okay, a nucleotide has 3 parts to it: a sugar, a phosphate, and a nitrogen base. Okay, sugar, phosphate, and nitrogen base. So our monomer has its own little parts. So you can see those here on this picture. You can see my ring here. There's my sugar. Okay, here's my phosphate. And here, over here, is my nitrogen base. Most of y'all remember, as you talk about DNA, your A to T's and C to G's, those are your nitrogen base. Okay, so our monomer is the nucleotide. You can see over on the left here, I've got a bunch of these nucleotides all strung together. So there's my individual monomer, bunches of them, and so they're linked together into one big polymer. And so in this case, my polymer is also called my nucleic acid, and I have 2 examples of these, DNA and RNA. Okay, and we're going to talk a lot about each one of those individually. So our functions of our nucleic acids are going to be to store genetic information. In particular, DNA is the one that does that. Here's DNA over here on the side. Okay, remember our double-stranded A to T, C to G. You can see your nucleotide pieces here, the phosphate, the sugar, the nitrogen base, all of that making up the nucleotide monomer. All those nucleotides together make up the nucleic acid polymer. Okay, in addition to storing genetic information, nucleic acids are also able to transmit information, and they help make proteins. Synthesize or make proteins, which are important, especially those enzyme proteins. And this would be the job of RNA, which is going to help transmit and synthesize those proteins. And the last thing we need to mention here is what's called ATP. Okay, ATP is a modified nucleic acid. So it's not, actually it's a modified nucleotide. So it's not going to make up DNA or RNA. ATP is extremely important to us because ATP is our source of energy. It's our usable, a lot of times we refer to it as our usable energy source. Okay? And it's called ATP because it's an adenine okay, so 3 phosphates, okay, adenine triphosphate. Okay, and so it is a modified nucleotide because it's got our sugar, it's got your nitrogen base, okay, but instead of having just one phosphate group like most of them do, it's got the full 3 here, which makes it a modified nucleotide. But it is our usable source of energy, and so it is extremely important to us. Okay, so be sure that you've gotten all of your notes regarding all of this information. We're going to be doing a big lab covering this in class. We're going to be making some foldables, and we're going to be doing some more stuff to help you remember all of this.